First of all, I would like to thank the organizers of this interesting conference to let me participate with the talk, Influence of Sex and Diet on the Gastrointestinal Tract in a Mice Model with Partial Deficiency for TGF-beta-3. This research has been performed at University Rey Juan Carlos, Spain, and I will present it on behalf of my co-authors, Paula Gallego, Ana Baués, Elias Casani, Yolanda López Tofiño, Antonio Márquez, Gemma Medina Gómez, and José Antonio Uranga. Transforming growth factor beta-3 is a member of the family of cytokines transforming growth factors beta, TGF-beta, which are involved in a wide range of biological processes important for homeostasis. In fact, the dysregulation of these cytokines has been related with the occurrence of different diseases, like cancer or chronic inflammatory diseases. In particular, TGF-beta-3 has been shown to be key for adequate craniofacial, lung, and cardiovascular development, and mice completely deficient in this protein display cleft palate, among other alterations like altered heart development, and die shortly after birth. In contrast, heterozygous mice for this gene survive normally, with no apparent external differences with wild-type litter mates. However, heterozygous mice display some alterations. For example, Petrus et al. Have found that even if these mice grow normally as their wild-type litter mates fed with a control or with a high-fat diet, the mutation in high-fat diet-fed animals increased the size of adipose cells in the subcutaneous adipose tissue, not their numbers. As you can see here, these were decreased instead of not modified. Thus, TGF-beta-3 seems to play a role in adipose tissue expansion by hyperplasia associated with obesity. In addition, in these heterozygous mice, at the embryonic age, both heart and vessels have been shown to display some mild lesions, whose impact in adulthood is still under investigation. Thus, the question is, what is the role of TGF-beta-3 in other systems? And in particular, we ask, what is the role of TGF-beta-3 in the gastrointestinal tract? It is known that overexpression of TGF-beta-1, another member of the family, promotes spontaneous colitis and fibrosis, and this may contribute to the development and maintenance of inflammatory bowel disease. Inflammatory bowel disease is a severe inflammatory chronic immune-mediated disease of the intestinal tract with two main types, Crohn's disease and ulcerative colitis, as you can see in this figure. It is well known that in both kinds of inflammatory bowel disease, gastrointestinal motor function and gut wall structure are altered. In particular, in Crohn's disease, any part of the gastrointestinal tract may be inflamed in a discontinuous, patchy manner, and the full thickness of the bowel wall is affected. That means that inflammation is transmural in these patients. On the other hand, in ulcerative colitis, the characteristic is the presence of inflammation from rectum to more colonic, proximal colonic areas in a continuous manner. And in addition to this, only the mucosal and submucosal layers of the colon wall are affected. Both types of inflammatory bowel diseases are dependent on age, sex, and environmental factors, particularly diet. Thus, incidence of Crohn's disease is higher in teenagers and middle-aged women, whereas ulcerative colitis is more prevalent in men older than 45 years old. In addition, diet-induced obesity is an important risk factor for inflammatory bowel disease development. So, taking all the mentioned factors into account, we may also ask, do sex and diet affect TGF-beta-3 role in the gastrointestinal system? So the aim of our study was to determine the possible alterations in motor function and structure of the gastrointestinal tract in mice partially deficient in TGF-beta3, and to determine the influence of diet and sex in these alterations. In our study, we used 4-week-old mice of both sexes. Some of them were wild type and some heterozygous for TGF-beta3. The mice were fed either a control, standard diet, or a high-fat diet for 12 weeks. Therefore, we had 8 groups, 4 of females, 4 of males, depending on the diet and the genotype, with 11 to 16 mice per group. After 12 weeks of feeding, the gastrointestinal motor function was evaluated in 2 phases. In the first one, a suspension of barium, which is a radiopaque substance, was intragastrically administered and mice were placed in individual cages without bedding or food, but with water. Fecal pellets were collected, counted, and weighed both before and after drying in an oven, so that the percentage of fecal moisture could be calculated. In the second phase of the study, the feces were radiographed. As you can see here, these are the fresh, the real feces, and these are the radiographs for these feces. And different parameters were obtained to evaluate the possible changes in gastrointestinal transit. For example, we calculated the stool marking index at each time point so that curves could be built along time for this parameter. In addition, we recorded the latency of expulsion of the first labeled pellet, which in this example happened at 2 hours after barium administration. After the functional study, the animals were sacrificed by quick cervical dislocation and the different gastrointestinal organs were weighed and their size for stomach and cecum or length for small intestine and colorectum were determined. Finally, we performed a histological analysis of the distal colon to determine the possible alterations in the colonic wall structure and on the submucosa thickness using Mason's trichromic staining. And now I will show you our main results. Regarding the first phase of the functional study, we found that in wild-type animals exposed to controlled diets, both males in purple and females in black, expelled relatively more fecal pellets at the beginning of the study, but the number decreased along the session. This could be simply due to the effect of the stress of the animals facing a new environment at the beginning of the study and posterior habituation. This pattern was not significantly modified by the mutation in either sex, but the high-fat diet, which is blue in males and red in females, decreased the number of pellets expelled at the beginning of the session in both sexes, making this parameter a bit more constant or stable throughout time than in animals exposed to controlled diet. Interestingly, the dry weight of the fecal pellets was also quite constant along time in all animals, suggesting that the initial difference in the number of fecal pellets had to be due to a difference in moisture. Indeed, the dry weight of the, when we evaluated the percentage of moisture, we found that this parameter was highest at the beginning of the study, but decreased a long time, as shown for the number of pellets here. This happened in wild-type animals, male and female mice, and also in heterozygous male and female mice exposed to either control or high-fat diet. Interestingly, only high-fat diet produced some differences with control animals, with the amount of moisture being less than in animals fed the control diet, particularly in females, as you can see here. This is somehow an expected finding, which could just be explained by the physical properties of fat. Retention of water in feces might be simply more difficult when these are more fatty, due to the higher fat excretion in animals fed the high-fat diet. The curves shown in this other slide illustrate how barium intragastrically administered reached the feces a long time. To understand this second phase of the functional study, it is necessary to remember that mice do not vomit, and therefore barium can only travel distally within the gastrointestinal tract. In general, we observed that, as expected, the stool marking index increased a long time in all animals, in this graph and also in these other graphs. Regarding the effect of sex alone, although the differences were not statistically significant, it seemed that barium reached the feces a bit faster in males than in females. This might be related with females displaying constipation more frequently than males. When we evaluated the effect of the other factors in males, we observed that the genotype of the diet did not significantly alter the curves compared with those obtained in wild-type mice exposed to the control diet. However, in females, it seemed that the high-fat diet in red accelerated the occurrence of barium in the feces, particularly in the heterozygous mice, which had the continuous line. Then we decided to evaluate a bit more precisely how transit was modified by looking at the latency of expulsion of the first marked pellet. In this table, we can see first that barium-stained fecal pellets occurred at a significantly longer time in females than in males. This is in minutes, further supporting that females showed a slower transit than males. Curiously, fat tended to make latency similar in both sexes, meaning that fat slightly increased the latency in males and slightly decreased this parameter in females, although the differences were not statistically significant compared with the corresponding males and females fed a control diet. Interestingly, the mutation alone significantly increased the latency in males, but did not modify it in females. Furthermore, when mutated animals were fed the high-fat diet, the latency was normalized in males, meaning that this diet counteracted the effect of the partial deficiency in TGF-beta3 in male mice. In contrast, the high-fat diet did not significantly alter this parameter in mutant females. Next, we studied if there were any macroscopic changes in the gastrointestinal organs associated with sex, diet, mutation, or the combination of factors. For this, we first spread the gastrointestinal tract on a piece of paper that was used to measure size or length of the organs with the aid of ImageJ free program. We analyzed the size of the stomach and the cecum, as well as the length of the small intestine and the colon. We also recorded the weight of these organs separately, but without removing their contents. The first thing we noticed is that appearance of the organs was normal in all animals, irrespective of sex, diet, or genotype, with no sign of overt inflammation suggestive of the development of inflammatory bowel disease. Although we could observe some minor changes due to sex, like here, or mutation alone, like here, in the small intestine or colon weight or length, we saw many differences induced by high-fat diet in wild type or in heterozygous mice. Thus, in wild type and heterozygous mice fed a high-fat diet, the gastrointestinal organs were smaller and shorter or weighed less. This was especially remarkable for cecum and colorectum in both sexes, whereas differences were evident in the stomach and small intestine only for females and only for their weight. It is known that inflammatory bowel disease is associated with colon shortening. Maybe the macroscopic changes found in our study could be associated with the occurrence of the low-grade inflammation typically associated with obesity, which was actually present in animals fed the high-fat diet. Thus, one could expect that due to this reduction in size or length, gastrointestinal transit was faster in all high-fat diet-fed animals. But as previously shown, transit was not significantly altered by high-fat diet alone. Thus, the microscopic changes shown here do not seem to have impacted transit in our animals, although of course it cannot be discarded that a longer duration or high-fat diet exposure might provide different results. On the other hand, the slower transit found in mutant males that was normalized by high-fat diet cannot be explained either by changes in the microscopic appearance of the organs. Finally, we looked at the colonic wall's histological structure and confirmed that it had a normal general appearance. Thus, irrespective of sex, genotype, diet, or the combination of these different factors, we did not observe the typical features associated with inflammation like ulcers, goblet cell depletion in the mucosa, or inflammatory infiltrates. So the appearance of the gut wall in all animals was normal. However, when we measured the submucosa thickness, significant changes were observed in animals heterozygous for TGF-beta3 and exposed to control diet. In particular, submucosa thickness decreased in males and increased in females in comparison with their corresponding wild-type litter mates, as you can see here. These are very interesting results, suggestive of a role of TGF-beta3 in line with that of TGF-beta1 in the colon, whose overexpression, as mentioned in the introduction, is associated with the development of colitis and fibrosis, at least in male animals. So if there is a reduction here, it could be precisely because of the lack of a part of the protein needed. The different effect of the mutation depending on the sex of the animals is also clearly an attractive finding that needs to be more deeply studied. So to conclude, no experimental group showed signs of inflammatory bowel disease. Wild-type females presented lower body weight and delayed gastrointestinal transit compared to males. High-fat diet counteracted the initial stress of animals during the X-ray session, particularly in females. The partial deficiency of TGF-beta3 produced different effects depending on the sex of the animals. So in males, it reduced both gastrointestinal transit and submucosa thickness. And the question is whether this is similar to the role, if the role of TGF-beta3 is similar to the role exerted by TGF-beta1, whose overexpression favors IBD development. In females, in contrast, the mutation in TGF-beta3 did not alter GI transit, but increased submucosa thickness. And the question is whether mutant female mice could be more prone to IBD development. And finally, the high-fat diet normalized gastrointestinal transit in mutant male mice, but tended to increase submucosa thickness even further in female mice. And the questions here are, what are the mechanisms involved and what would be the implications for these findings? Finally, I would like to acknowledge the technical assistance provided by Laura López Gómez and Adrián Sánchez Yáñez. And we are, of course, also thankful to the funding agencies that have provided the resources for this research. And in the current pandemic situation, I would not like to finish my presentation without expressing our solidarity with those who are or have been ill with COVID-19, the families who have lost members, the healthcare workers on the front lines of this pandemic. And thank you very much for your attention.